Hello, I'm John Newmeyer. Welcome to the Grand Rounds for the Department of Psychiatry and Behavioral Sciences. I'm filling in for Dr. Joseph Ceramelli today. Couldn't be here today. And our Grand Rounds program is sponsored by a group of our staff who have done a wonderful job of organizing a hybrid today where we're both in person and presenting on the web. And so I want to thank Semhar for working on the communications aspects, and Annalie and Rosemary for working on the presentation today. This program today is being sponsored by the Garvey Institute for Brain Health Solutions. And next, we'd like to introduce our speaker, Dr. Kerry Ressler. I'm only going to describe his achievements briefly because it would take the full time of the presentation otherwise. But Kerry graduated from MIT, and then he attended the medical scientist training program at Harvard. And he did his PhD dissertation work with Dr. Linda Buck, who later became a Nobel laureate and also a UW faculty member currently. And then he began his career in psychiatry as a resident at Emory and did his fellowship with Michael Davis, who's had a great impact on me over the years, and then Kerry as well has done so. He's had over 500 publications, and his H-index is 130, which is almost 3 times mine. He's very active in professional societies. He's the past president of the ACNP, the American College of Neuropsychopharmacology, and the Society for Biological Psychiatry. He's also a member of the National Academy of Medicine. He's also the James, and he's, after rising through the ranks at Emory, he returned to Harvard, and he's now currently the chief scientific officer at McLean Hospital. And he also occupies the James and Patricia Poitras Endowed Chair in Psychiatry. We've worked together a little bit in the past at the Neuropsychopharmacology Reviews when he was a guest editor for one of the issues, which is a tremendous amount of work, and he was able to do that really efficiently on top of everything else. I think that characterizes his effectiveness and his outreach to many different audiences. His research spans genetics, basic and clinical neuroscience of fear, and also the public health impact of trauma. He's contributing to our understanding of the effects of trauma, focusing both on the internal, the individual biological and psychological processes that occur, but also the impact of external factors induced by society and environment. His work envisions improvements in existing treatments and development of new therapies. So Kerry, thank you very much for presenting today.
Thank you. Thanks so much, John. Really appreciate the kind introduction. And in addition to all of your great science, you're also a great comedian, so thank you. All right, so thanks to everybody who made this possible today. I'm delighted to be here, and I look forward to talking with the residents afterwards as well. So I'm going to tell you about, spend about the next 45 minutes walking you through a variety of approaches to how we understand post-traumatic stress disorder, both from the epidemiological scale of the disorder, the symptoms of the disorder, how it's comorbid with many of our other disorders, but also try to make the argument that understanding trauma is tractable and feasible, and there may be some very exciting future approaches not that far away from our understanding of the neurobiological mechanisms of threat and fear to novel interventions and preventions. So with that, I will jump right in. So first of all, in preaching to the converted here, you all know that serious mental illness, including PTSD, are the major causes of disability worldwide, both in terms of it's even worse now. This was 2011 World Economic Forum that mental illness globally will cause more than cancer, diabetes, and chronic respiratory diseases combined, and in terms of lost individual lives, daily lives. Sandra Galea of the BU Public Health has stated trauma is a foundational driver of the health of the public, and that mental disorders broadly and trauma also broadly are two of the top 3 components that are the 10 medical conditions with highest estimated spending. On that note, I think, as we all appreciate, the amount of federal spending, NIH spending, et cetera, in mental disorders is much less than many of these others together. And the points that I'm going to try to make is, one, from an epidemiological perspective, we have to do more, but these are also solvable disorders. If we put the money into the research, we can have really powerful novel approaches, just like cancer was transformed over a 30-year period in infectious disease by putting money into understanding the biology. This is sort of my clinical DSM slide all in one place. So, so we'll start with just reminding you that that post-traumatic stress disorder, PTSD, is clinically important. It affects about 5 to 10% of the population. As I talk through this, the round number you can have in your head is about 10% of people after a very severe trauma may go on to develop PTSD. However, after multiple traumas or multiple amounts of trauma loading, as it will, those numbers increase. And we see combat veterans and at-risk populations. A lot of our work has been in inner city Atlanta in minoritized populations with chronic stress, chronic structural racism, and lots of exposure to violence. And in our at-risk community populations and our at-risk soldier populations, we can see rates of PTSD as high as 25%. So it is prevalent. It is one of the highest risk factors for suicide amongst our disorders. It is one of the most common. I'll show a comorbidity slide. Up to a third to a half of people with PTSD have comorbid depression or substance abuse. And so there's many reasons why we need to focus on this more. As in terms of diagnosis, what you'll remember is that the first month after a severe trauma, we don't call it PTSD, we call it acute stress disorder. And the reason that is that most people, those first days and weeks after trauma, are very upset, have difficulty sleeping, are easily startled, can't stop thinking about it. You have many of these same symptoms, but most people will recover over time. And we'll talk about the natural recovery. So we generally, and by consensus, the diagnosis of PTSD requires a month of symptoms following trauma, and then chronic PTSD with more than 3 months. As a medical student, you know, we like mnemonics. So I'll leave you with a mnemonic today for what are the criteria. So errands, criterion A is the event, B is re-experiencing, C is avoidance, D is negative alterations and decreased mood, and E is sympathetic arousal. So again, for A, while we live in sort of a TikTok world in which everybody's been stressed or traumatized at some level, and certainly there's a lot of distress in the world, when we talk about PTSD clinically, we really talk about very severe trauma exposure, people who either themselves were nearly lost their life or saw somebody else lose their life, or being assaulted or sexually assaulted. And importantly, this response involves extremely intense fear and helplessness. In terms of the symptoms, again, that one needs to have for up to a month to 3 months afterwards, is the first is re-experiencing. This is the classic nightmares and flashbacks. This is interesting in terms of trying to separate when you're diagnosing people, you know, depression with stress versus real PTSD. Criterion B is probably the most specific. Are these people being haunted by a trauma memory that they can't get out of their head? They essentially feel that, you know, this is a black hole of emotion kind of pulling them down. And then in terms of the sleep disruption, much of that's driven by the hyperarousal, but also with the nightmares of re-experiencing this. And even with severe PTSD, just like severe depression, you can see pretty significant psychotic symptoms. And a critical thing to ask about is the same way if you're looking at affective psychosis, is it mood congruent? With PTSD and psychotic symptoms, is it essentially, are they being haunted by these memories? Are the voices they're hearing somebody of their attacker, et cetera? And then criterion C is avoidance symptoms. And with all anxiety disorders, we see people avoid. And of course you avoid because you don't want to be doing the thing that scares you, but that also tends to make the disease worse and prevents recovery. And it also is part of what really gets in the way of functioning, whether it's not being able to leave their house, keep a job, have a relationship, et cetera. Negative alterations in cognition and mood. This was added from DSM-4 to DSM-5, and it's really trying to capture both the deficits in executive functioning, the inability to cognitively be flexible and cognitively have executive functioning when all of your brain is focused on survival, as well as sort of the numbing and anhedonia that we see that looks very much like depression, but with people with chronic trauma. And then finally, the sympathetic hyperactivation, the decreased sleep, the startle, hypervigilance is the other component of PTSD that is relatively specific. This is great. If you want to hear more stories, the VA has put a really good site together called maketheconnection.com. I'm not going to have time today, but literally hundreds of videos of people telling their stories. You can type in any symptom cluster and you can hear somebody's stories of that startle or re-experiencing or nightmares or recovery. And it's very useful for patients as well. In terms of the comorbidity, again, trauma, as Sandra Galea said, it's one of the foundational drivers of public health and mental health. Again, almost 50% comorbidity with depression, about 30 to 40% comorbidity with substance abuse, and of course, the whole range of anxiety disorders. In addition to to psychiatric disorders, what we see with PTSD is really a lot of physical and somatic symptoms. And this brings us back to the point that historically, we did not define PTSD as a disorder until really the 70s, after the Vietnam War, for the military component, and then really after the women's rights movement and appreciation of the rates of sexual trauma in the 70s. But the concept of PTSD exists as long as there has been written history. The ancient Greeks and Romans talked about people who were forever changed after a fire or a war, and some of the terms after the Revolutionary War and the Civil War and World War I in the America are things like soldier's heart, irritable heart, shell shock, combat exhaustion, battle fatigue, nervous shock. These were all symptoms that when you read about them are essentially exactly PTSD. And we know PTSD, like depression and other severe mental illnesses, have much higher rates of cardiovascular illness, cancer, et cetera. We are starting to understand mechanisms of this. Certainly the circulatory, cardiovascular, and sympathetic and parasympathetic alterations of trauma are associated with cardiovascular risk. We've recently shown in about 100,000 person polygenic risk score study that there's shared genetic risk of PTSD and cardiovascular risk. And then we think that the inflammatory and redox effects that you see in metabolic dysfunction you see in PTSD are also associated with increased cancer risk. Importantly, PTSD I think of as sort of the perfect illness of gene by environment. We know that everything in psychiatry and many things in medicine are really nature and nurture, or gene and environment. But rarely is there a place in psychiatry where we understand the environmental component as much. By definition, with PTSD, it is that trauma. And everybody who has studied PTSD since we first started talking about PTSD has studied partly what that trauma is. So that gives us a handle on that environmental component that is easier to study than perhaps other areas where we know there's a component, but it's harder to break that down. And I'll be talking more later about the genetic component that's made a lot of progress in the last 10 or 15 years. I also just wanted to talk a little bit about civilian trauma. So while I'm going to sort of go back and forth between PTSD, military trauma, civilian trauma, and animal models of threat and fear, the area for which I've most been involved and contributed to in the area of civilian trauma is what we call the Grady Trauma Project from my 18 years in Atlanta. So in about 2000, we started this project. I was with Beck Bradley and Tanya Ivanovich and others in the early 2000s. We were either residents or we were shortly out of residency, and we recognized in Grady Hospital, which is one of the largest remaining charity hospitals in the country, that everybody coming into our psychiatric emergency room or our county mental health center, first of all, as a resident, as many of you may know, nobody seemed to perfectly match DSM. I felt like I was a horrible diagnostician because everybody seemed to have everything. And that may partly be true, as we all understand, and they're trying to deconstruct nosology to some extent now. But what I also saw was that everybody seemed to have trauma, and that it was a component that we really weren't asking about in the 90s. So we started studying this first in our mental health center, and then we started working with undergraduates and volunteers and doing interviews in the general pediatrics clinic and general medical clinic and waiting rooms and every other part of the hospital we could for people who are not seeking psychiatry. And over the course of about 15 years, we interviewed 13,000 people. It was about 95% African-American because that's where Grady is located in an impoverished part of Atlanta. And it takes you don't have to pay anything to go there. It takes Medicaid, whatever. So essentially, everybody who is in the Grady population that don't come for trauma to the emergency department is often under the poverty line. And so there's an extreme poverty, but the rates of trauma were just enormous as we gathered more epidemiological data. Almost 50% of the people in inner city Atlanta know somebody personally who's been murdered. Almost half have been personally attacked themselves. Almost a third have experienced sexual assault or childhood maltreatment. And while these numbers are horrible, they're not unique to Atlanta. We see the same thing in poor parts of New York and Detroit and Chicago and LA. I know Portland has components of it. So it's really epidemic problems of impoverished America and often highly minoritized aspects of America. And what we've also increasingly appreciated is that high levels of experienced discrimination for decades and generations also contributes to the chronic stress that increase risk. So when we think about what are the things that increase risk, they're trauma exposure, they're low resources, but it's also chronic stress. And we increasingly appreciate that chronic structural racism is part of this. So there's been over about the last decade, a lot of studies now that sort of have grown out of the Grady Trauma Project, led by these three colleagues on the right, Nagar Fani at Emory, Nate Harnett at McLean, and Sierra Carter at Georgia State. And they've really done some terrific work in this primarily minoritized population to understand structural inequities, psychological inequities, neurophysiological components, neuroimaging components, and then also working with cohorts like the ABCD consortia for developmental brain effects to understand the roles of structural racism. So I'm really proud of the Grady Trauma Project, both for helping us better to understand the quantification and impact of trauma, to help us with the biology that I'll be telling you about. It was one of the founding cohorts in the Psychiatric Genomic Consortium for large-scale genetics, as well as large-scale imaging, but also is really helping us to better understand the effects of structural racism and risk in trauma. I'm going to not talk a lot about current treatments today. This is my main slide, but very happy during the discussion to talk about how to treat patients or how to deal with difficult situations. I am a psychiatrist and treat patients as well as do research. As with most things in psychiatry, we have two paths to follow. We have medications and we have therapies, psychotherapies. The psychotherapies in PTSD have the best evidence. And you can basically think of this as the medications that we use. First of all, start on the bottom. We currently have only 2 FDA-approved drugs for PTSD, and that's sertraline or Zoloft and paroxetine or Paxil. So again, old antidepressants are all we have that the FDA is currently approved, which is just very sad. You know, I've said all the reasons why this is an important disorder. What we now know is that the broad range of drugs that we use for depression and anxiety work for PTSD as well, SSRIs, SNRIs, tricyclics. Benzodiazepines we try to use very sparingly for several reasons. First of all, because of the tolerance issues, because of the comorbidity issues with substance abuse with PTSD, but also there's evidence that this can interfere with the exposure component of recovery that I'll tell more about. I won't talk much about prazosin, but it's probably one of the most interesting drugs that we have. And again, that came from the Seattle and UW system with Murray Raskin. And it's a very interesting drug. It blocks the adrenergic, alpha-1 adrenergic system. It seems to, when used properly for the right patient, really help with nightmares and help with hyperarousal. So of our drugs, it's probably the only one that's really specifically targeting one of the systems we know is dysregulated. The problem with prazosin is the therapeutic window between when you actually get orthostatic and with low blood pressure versus when it really helps with nightmares is very short. So you really have to titrate it very specifically, and every individual is different. But it's potentially a very good, at least initial treatment in that space. But again, there's no currently FDA-approved treatments based on our understanding of the neurobiology. So that's what I'll spend a lot of the rest of the time today talking about, is how our neurobiology can lead us to new treatments. In terms of the therapies, all of the well-defined therapies with really good effect sizes are based on exposure ideas, the idea of trauma exposure. And prolonged exposure is the one that's really the best understood. Dialectical behavior therapy, that is again another product of UW, it now has a DBT PE component, and they're using DBT for a lot of skills training and emotion regulation doing the prolonged exposure. There's arguments in the field about how much you can go straight into prolonged exposure. People like Edna Foa would say you can go straight into it with everyone. Many people would say you're much more likely to see dissociation or avoidance or a lot of emotional dysregulation if you don't have skills training and emotion regulation first. And then there's things like EMDR that can be very helpful. Eye movement desensitization and reprocessing is essentially a form of exposure therapy that has a very strong grounding component, whether you're watching something and moving your eyes or tapping. And I think this preponderance of evidence suggests that there's probably not anything particularly powerful about the intrahemispheric component with EMDR, but more the externalized grounding that keeps somebody very grounded and focused and prevents them from dissociating. I'm not going to talk much about dissociation today, but whenever you're thinking about subtypes of PTSD, probably the very first thing to look at is sort of complex PTSD with dissociation versus more classic PTSD without dissociation. They seem to have very different neural circuits and probably different ways of responding to some of these treatments. So happy to talk more about that, but I want to talk for the rest of today about where we are as a field for the future. And the first point is that in addition to PTSD being important clinically and epidemiologically, I think PTSD may be one of the most tractable disorders that we have. And I would argue from a neurocircuit perspective, the addictive disorders, addiction and trauma and fear disorders are two of the ones for which we have the best understanding of the neural circuitry of these behaviors. And importantly, the circuitry that is shared really across mammals and even vertebrates in terms of the threat and fear system that we can think of as dysregulated. Second, PTSD, we know when it starts. It's relatively unique in psychiatry. By definition, we know when it starts at the time of the trauma. That gives us a lot of power for both studying it, but also for identifying people right after trauma and maybe intervening and preventing it. And as I talk about future directions, one of the real sort of visions is in the same way that right now, if you have arm pain or chest pain or arm numbness, you go to the emergency department and with an EKG or with an EEG or with a CAT scan and with blood chemistries, you can say, yes, you're within X number of hours of having a myocardial infarction. We can do this intervention and we can prevent the long-term sequelae of a heart attack. If we knew right now that you were 95% likely to have PTSD after this trauma, we could apply some of these new interventions in those first hours or first night of sleep and prevent the PTSD from ever developing in the first place. We have the knowledge now to start to be able to do that. How do we refine that and implement it for the emergency department or the battlefield? And then finally, PTSD really is built on this concept of learning and memory. We often think of learning disorders as dementia, for example, or cognitive dysfunction. But PTSD is really a disorder where we've overlearned, over-encoded that initial trauma memory, and you now can't modulate that memory as well. But learning and memory is one of the areas in neuroscience that's made the most progress in the last decades, and how do we implement that? So our goal then is to have a molecular neurobiology of PTSD based on neural circuitry for predictive biomarkers and novel interventions. So before I go into some of the new experiments, just one more bit of background for thinking about the neural circuitry of PTSD. So I like to use Joe Ledoux's classic, I guess it's like a 30-year-old figure now, and it shows a guy walking through the woods. And I guess the Pacific Northwest may have some snakes. Where I lived in Atlanta, there were lots of snakes, and my wife's deathly afraid of snakes. So I always like to talk about going through, we'd go for a walk down the road, we'd be having a nice conversation, and then Betsy would be jump behind me. Yes. And I'm like, what's going on, Betsy? And she'd say, it's a snake. I'm like, where? She says, there. It was a stick. It was always a stick. And so Betsy's amygdala was prepared for the snakes of the world to come and get her, right? So we can all kind of laugh about that, and everybody's got their own story. But that's the way the fight or flight system works, right? And so what's happening at the next level is all of our sensory systems are constantly online, tuned into the world around us. And in terms of the visual system, the eyes, the ganglion cells from the eyes send in one synapse to the visual thalamus, the lateral geniculate, and then that sends projection to the primary visual cortex, the secondary visual cortex, V4, MT, higher order association areas, and eventually prefrontal cortex. Well, that takes about 500 milliseconds, half a second, for us to essentially be consciously aware of what our sensory system saw 500 milliseconds ago, which by itself is kind of a philosophically strange idea. We're never really living in the moment. We're always living a second or two ago, right? In terms of what we're aware of. Where the amygdala receives this pre-processed information and in about 100 milliseconds fires off the reflexive fight or flight system. The amygdala is constantly comparing this pre-processed sensory information in real time to memory engrams, if you will, of prior threat information. So now we live in the system where the amygdala has led us to survive. It's led us to run away from the lion, to be afraid of the snake or whatever, activate a whole set of fight or flight or freeze responses. And then we only afterwards we realize that we were overly upset. Well, people with anxiety and threat disorders are living with this all the time and they can't shut it off. So we can see why it's a primary driver of survival. And again, the amygdala, as we know it, is essentially the same in non-human primates, very similar in rodents, and still quite similar in vestigial in reptiles. So it's a primary driver of survival, and yet we didn't evolve for a world with 24/7, trauma news cycles, with social media, trauma and news cycles. And see, why we have sort of such an anxious world right now, because we evolved for a different world than we're currently living in. But with people with PTSD, they're constantly on this fear system and they can't turn it off. So at the next level, we can think of the amygdala as constantly receiving the sensory information from the conditioned stimuli, maybe the color of a car or someone's face that was previously neutral. But after the trauma, the car crash or the assault, that now is encoded in the brain, in which now the neutral stimuli activate all the same hardwired systems as the trauma itself did, the unconditioned stimulus. And we know that this learning occurs very rapidly, involves mechanisms like long-term potentiation, synaptic plasticity, it's NMDA dependent, it's BDNF dependent. A lot of that's been worked out over several decades. What is also known and was figured out really in the 70s and 80s was the amygdala has hardwired projections to a whole host of brainstem and subcortical regions that activate all the symptoms that we see with a panic attack. So if you activate the amygdala chemically or electrically, you get the downstream effects of increased heart rate, GI distress, respiratory distress, arousal, startle, and then the corticosteroid release from the hypothalamic pituitary adrenal axis. So almost no place else in psychiatry do we really have this behavioral reflex that we understand at a neural level, really through these specific synapses that come from this very rapid information. And it's constantly being modulated by the cortex and hippocampus, but for those with PTSD, they can't turn it off. So I think it's helpful to break down the concept of PTSD as not one thing, but multiple things at multiple different time points. So again, if we think of a single trauma, one has about a 5 to 10% chance of developing PTSD versus about a 90% chance of recovering. So what's different between those two? Well, first of all, there's the idea of pre-existing sensitivity. This is where gene by environment comes in. And I'll show you data in a minute that we know now about 30 to 40% of the risk is genetically heritable. And we're making progress on what those pathways are. But we also know that prior existing environment, so childhood trauma, we know is one of the biggest risk factors in all of psychiatry, but we also know that particularly in PTSD, childhood trauma likely sensitizes these amygdala systems so that later adult exposure to trauma is built upon an already sensitized threat pathway. The event itself matters. We know that interpersonal trauma is a higher risk factor for PTSD than non-interpersonal trauma. We know that having things like social support, having Maslow's triage of financial resources, food resources, all of these sorts of things, and social support are all components that help protect people, as well as emotion regulation. We know that a history of anxiety disorders and other psychiatric disorders increases risk and probably is increasing stress sensitization. Then there's the concept of consolidation, that memories aren't formed immediately, but formed over minutes to hours to days, and that there's very likely biological and psychological and environmental components that are different there, but we think that's a critical point for intervening to prevent long-term PTSD. And then in these first weeks after trauma, that period where you have normative stress or acute stress, that you transition from acute stress to PTSD versus recovery, a lot of these symptoms can further exacerbate systems. For example, the intrusive memories, the nightmares, the flashbacks, the avoidance behaviors, the sympathetic response, they can drive different cognitive responding that then leads to recovery or PTSD. So for example, with general concept of threat generalization, someone will say, I was attacked down a dark alley by this person at night. And at first I was just afraid of that situation. Then I didn't really want to go out at night anymore. I was kind of afraid of men after that. And then I didn't want to go to that part of town. Now I'd rather kind of stay in my house. I'd rather just be in my bedroom. The world has become more and more dangerous and less able to carve out safety spaces. Those who recover are more likely to discriminate. I know there's dangerous places and safe places, but the whole world is not dangerous. Concept of sensitization is interesting. This is where people will say, doc, every time I talk about it or I have a nightmare, it gets worse and worse. Why would I want to talk about my trauma? And about 1990, I guess, Joe Ledoux's group restarted studying a process called reconsolidation. And the idea there was anytime a memory is reactivated, that it becomes labile and can be strengthened again. And we think what is happening, and I'll show you in some of our studies if I can move a little bit faster, is that anytime a memory is reactivated, it can either go down these opponent process of either be strengthened or reconsolidated or sensitized, or it can be inhibited and extinguished. And extinction is really the process of exposure therapy. Okay, so I'm going to talk about some of the large-scale genetic studies contributing to gene by environment, as well as some of our understanding of fear consolidation processes, and then how understanding of exposure and extinction may lead us to new approaches to making treatments better for people who already have PTSD. So how can we understand those at risk and maybe intervene early after trauma, or if you already have trauma, how can we understand the neurobiology for improvement? So related to consolidation, again, memories aren't made permanent immediately, rather they go through a period of short-term memory that then isn't consolidated to long-term memory. And this goes from transient molecular changes through new gene transcription to new protein, to new synapses, to new structural changes in the brain. And then anytime a memory is reactivated, it can be reconsolidated versus extinguished. So I want to talk first about this concept of consolidation. So Jim McGaugh, who's at Irvine for decades, really was one of the first in the threat memory field to talk about memory consolidation, both at the hippocampal and amygdala level, with the idea that any short-term event over the course goes from seconds to hours, short-term memories to long-term memories to long-lasting memories. And many of these things were replicated even in aplesia with Eric Kandel showing the early components of memory. In Michael Davis's group, we showed in the early 2000s that with amygdala fear conditioning and threat memories, you could see these same timelines of patterns in amygdala and could find genes that came up very early after fear conditioning, others that came up later and stayed later, including growth factors and structural proteins, and others including GABAergic genes that were transiently decreased, consistent with a transient excitability. So how do we study this in humans? So about 15 years ago, we started a study at Emory and Miami in emergency departments, identifying people right after trauma, collecting blood, and then collecting a whole component of physiological measures. And then the data from that led to a large-scale UO1 project led by Sam McLean, emergency department doc out of UNC, and Kirsten Conan and Ron Kessler, myself at Harvard. And this ruled about 14 or 15 emergency department sites and about 55,000 people. And we essentially, in addition to the immediate blood-based biomarkers and emergency department measures, we were able to do wearable experimental watches for actigraphy, for sympathetic and parasympathetic measurements, for sleep, as well as active and passive phone data, as well as bring people back from imaging. So I'm not going to talk about all that today, a lot of it, but a number of papers have come out. I'm happy to talk about what we're starting to learning about different ways of predicting the trajectories of PTSD. One of the things we found early on was that the pathways that we know are involved in the systems processing of threat consolidation can be seen very early on in PTSD. So again, on the left, we're seeing the trajectories of PTSD symptoms. A lot of this is using the PTSD symptom scale or the PCL5, the PTSD checklist. And what we find is that many people don't have many symptoms and stay relatively low. That's part of that 90% recovery. Some people start very high, but over the course of a few weeks recover very quickly. But some people start high and stay high, others are middling, and some even get worse. But importantly, looking at amygdala activation, for example, to fearful faces in an MRI task, even 2 weeks after trauma, long before we diagnose PTSD, is already well correlated with PTSD symptoms 3 months and 12 months later. So we're already able to see the hyperactive fear and threat systems early after trauma, consistent with long-term chronic PTSD. In contrast, the hippocampus, as I've talked about, one of the areas that dampens the amygdala, provides the context for safety, memories, this context for extinction. And generally, we see in chronic PTSD less hippocampal activation, and people like Doug Bremner have shown that both PTSD and childhood trauma are associated with smaller hippocampal volume and decreased hippocampal function. Similarly, in about 2 weeks after trauma, on the y-axis, I'm looking at level of hippocampal activation to an inhibitory task in the scanner. And we see that high levels of hippocampal activation are associated with very low PTSD symptoms at 3 months and 6 months, and low levels of hippocampal activation can be associated with much higher levels of PTSD symptoms. So the basic sort of circuit triad of amygdala, hippocampus, and PFC are all starting to play their roles of too much amygdala activation and too little top-down regulation early after trauma. Some of the areas we're most excited about are new biomarkers, and this one was quite exciting early on. It didn't quite replicate for a reason I'll tell you in Aurora, and so we're trying to figure it out. But in our first study of about 50 people, we looked at galvanic skin response in the emergency department right after they'd been traumatized. So often people would still be like on a body board waiting for their next C-spine to be recovered, and they were bored and were willing to be in our study. And we would put a galvanic skin response on their fingers and say, what brought you into the hospital? And that simple question, the GSR, which measures sympathetic response, before and after that question, was highly correlated with PTSD 6 months later and seemed to be a highly predictive biomarker. In Aurora, for a number of reasons, they didn't want us to ask too much in the emergency department about the trauma itself. So we did the galvanic skin response while doing the demographics. That's kind of boring, and we didn't see much signal. So we're waiting to replicate this. But we think early adrenergic activation is also consistent with early trauma consolidation. Okay, so what about genetics risk? So again, I said about 30 to 40% of the risk. And just in the last few weeks, we've been able to show you this. The Nature Genetics paper came out this week. And so the Grady Trauma Project, along with the Nurses Health Study and the Marine Resiliency Study in around 2008 and 10, started the original Psychiatric Genomic Consortium PTSD Working Group. And 10 years later, we have over 1 million people combining UK Biobank, the Millions Veterans Project, and a bunch of other studies around the world. And so it's very exciting. We now have a genetic architecture of PTSD. And so what we're looking at is a genome-wide association study here with over 1 million people in about 200,000 cases. Each dot is a single ACG or T common variant. And again, the large peaks are highly significant gene loci associated with PTSD. So we have over 120 genes, over 95 loci. I'm not going to go through them all. But one I'll focus on in particular in a minute, chromosome 17 is the corticotropin releasing hormone stress axis gene, but we also find genes associated with synaptic plasticity, like here's NCAM1. We found a number of GABAergic genes, a number of glutamatergic genes. So we find a number of pathways that make sense, and quite a few of them lie in classic stress pathways or classic synaptic plasticity pathways. So it's exciting that both there's validation of molecular components that we understand, as well as a lot of new targets. My own personal bias on this, it's a joke that I don't think anybody thinks is funny but me, but with schizophrenia, which of course was about 5 to 10 years ahead in terms of the size of the samples, it took them about 150 genes till they got to the dopamine receptor, DRD2. It was sort of the first gene in GWAS that was associated with our current treatments. In PTSD, the CRH receptor was in the top 10. So it was validating that those of us who studied the stress pathway were onto something in terms of large-scale genetics. So why am I excited about this particular gene? So the CRHR1 receptor is in chromosome 17. It's the receptor for corticotropin releasing hormone. And the amygdala and the BNST are two of these classic threat pathways I talk about. They cause, CRH to be released from the hypothalamus. All right. My ability to go backwards. Oh, there we go. So they cause CRH to be released from the hypothalamus, causing ACTH to be released from pituitary, causing cortisol to be released, and that feeds back. We've known for a long time, and here are a couple of examples from our own group from the last 2 decades, that if you overexpress CRF in the amygdala, in this case it was with lentiviral vectors, you would see increased startle in mouse models, again, one of our classic measures, as well as a number of other anxiety-like behaviors. If you overactivate the CRF neurons, in this case it's a little more genetically difficult. We basically knock out the primary GABA receptor, GABA alpha 1, in the CRF neurons, so they fire more rapidly. Those animals show increased CRF expression, increased corticosterone, so increased HP axis, but importantly, they show increased fear and decreased extinction. So on tone shock pairing, in this case showing mouse fear learning, we see no significant differences in learning to be afraid. But then after they've learned this fear and consolidated it, the top line is the mice who knocked out GABA and have an overactive CRF system. They're more afraid and they don't extinguish fear like the controls. So just 2 of probably hundreds of studies showing CRF effects in this. One of John and my favorite genes is called FKBP5, and this is my only FKBP5 story, but it's just to remind you that there's this whole other story about this gene that is involved in the hypothalamic pituitary adrenal feedback system. The FKBP5 gene modulates glucocorticoid receptor regulation, but it's critical in the whole HPA story. And Elizabeth Binder, who's now the director at Max Planck in Munich, and our group and others showed a while ago that there are different genetic versions of FKBP5, which are associated with increased risk versus relative resilience, even in the presence of child abuse for PTSD. And the risk allele of FKBP5, which is associated with a more sensitive glucocorticoid response, is associated with more amygdala activation, less hippocampal activation, and epigenetic association of increased PTSD risk. So again, just 2 of the pathways genetically and physiologically associated with stress responding and PTSD. So this, again, lots of work. I've only touched on a little bit, but I want to leave you with the following model in terms of thinking about the gene by environment effects of risk for PTSD. That the amygdala is constantly online, comparing the external world with internal memories. And this, might be in part through these rapid sensory systems. And in activating these hardwired pathways faster than we can be consciously aware, it activates the fight or flight or freeze system, or it doesn't care, is relatively resilient, or even may say go towards that thing, right? And that's being modulated by the hippocampus and prefrontal cortex. But a whole host of risk factors, childhood maltreatment, poverty, violence exposure, and genetic risk together seem to prime this system. And one example is we know through Regina Sullivan's work that developmental cortisol early in postnatal mammalian development leads to a more rapid amygdala maturation and sensitization. We know that FKBP5 and CRHR1 are two of the genes that can be differentially expressed, leading to increased sensitivity of the cortisol system, and that genetic risk for increased sensitivity to early trauma may then lead to an amygdala that is more sensitized and always prepared for stress and fear as an adult. Again, this is only some ways of thinking about it, but at least is a way of thinking about combining these stories. So in my last 5 or 8 minutes, I want to shift gears. I've told you about how we think about development overall, how we think about the genetic components, the environmental components. But what about if you already have PTSD and need to inhibit fear? So this is a figure I showed during prolonged exposure at the beginning. And the basic idea is with all of our prolonged exposures for any anxiety-related disorder, the basic idea is you talk to the patient, and at the time you might ask them on a 100-point scale, how anxious are you? 0, I'm falling asleep. 100, I'm having a panic attack and have to leave the room. And as a good therapist, you want to take people, whether it's in vivo exposure, I'm afraid of spiders and I'm going to hold this spider, or it's in imaginal exposure, tell me about your trauma. I want you to be able to get as close to that 100 as you can. I want you to be upset, but I don't want you to be panicked. And we're going to hold you there, and we're going to talk about it, and you're going to talk about it again, and you're going to talk about it again. And the goal is after, 30, 60 minutes of talking about that trauma, it doesn't have so much power over you. It's maybe a little boring. And then and so it settles down over time. You come back the next time, it's still upsetting, but not as bad. You do it again, you do it again. When that one becomes boring, you go to the next hot spot. That's what the ideal therapist with prolonged exposure therapy does. What we know doesn't help is if you activate that memory, you get upset and you leave. I walk out. I'm not going to talk about it anymore. I'm going to shut down. I'm going to dissociate. So we'll come back to that in a minute when we talk about resensitization or versus extinction. But this basic process of exposing a mammal to something they're afraid of and not having the unconditioned stimulus occur is Pavlov called extinction over 100 years ago. And when you do that, the animal or the person then shows less fear the next time. And we know extinction depends on the NMDA receptor. This is one of the main plasticity glutamate receptors. The amygdala is chock full of it. And Mike Davis's group had shown in the early 90s that when you extinguish fear, you're learning a new memory on top of an old memory that's a safety memory. So he trained rats to be afraid, or David Walker or Bill Falls, who's now the dean of University of Vermont. They trained rats to be afraid of lights. They would take rats, they'd put in a dark room. They would, every time a light came on, they'd foot shock them. And when you do that, it doesn't take very long for now the rats, when every time the light comes on, to be afraid. If you give a startle cube, they startle more. So the red line showing the rats after light shock fear conditioning. Then they extinguished them. They gave them 60 lights on, off, but no shocks. And then they brought them back. Now the animals aren't afraid of lights anymore. That's the extinction curve. They did psychotherapy for the rats. They're no longer afraid. But now they gave a drug that blocked the NMDA receptor only at the time of exposure. And then they tested them again. They were just as afraid as they ever were. They did not have plasticity during exposure therapy. They prevent them from learning the safety. So one of the early ways of modulating NMDA receptors was through the serine glycine modulatory site with a drug called D-cycloserine. And it makes the NMDA receptor work better. And so a decade later, we showed that if you gave rats a partial extinction, they would show some decrease in fear. But if you gave them that partial extinction with the drug that enhanced NMDA function, D-cycloserine, they showed much decrease in fear. So we could biologically enhance plasticity as the brain was relearning. So the reason we used D-cycloserine was because in the 60s, it was used for an anti-tuberculosis agent. The same cyclic moiety of serine blocked bacterial modification. So it was a safe drug and had been shown to enhance plasticity at a dose lower than its antimicrobial function. So we were able to show that if we, in this case with acrophobia as the first study, that if we took people with fears of heights and put them in a virtual reality for virtual fear of heights, acrophobia, with only 2 pills in a double-blind placebo-controlled study over 2 weeks, they would get much better, be much less afraid of fear of heights than the people who just got therapy alone. Normally, it would take 6 or 8 weeks of exposure therapy to have this level of improvement, and we could do it in just a week with 2 pills. And so with humans, we could make exposure therapy work better, just like we could in rats. And that was a very exciting time for the psychology field. And there were a bunch of studies, about 8 to 10 overall, that were positive in about a 5-year period. The Stefan Hoffman showing social anxiety improvements with dicycloserine, Adam Guastella in Australia, Sabine Wilhelm in Boston for obsessive compulsive disorder with exposure and response prevention, Decline and others in Europe with PTSD, and Michael Otto with panic. So it was a very exciting time. However, as more people tried it and different people were using different compounds and using different doses, et cetera, as with so many other drugs that are too good to be true, and pay attention to how good these effect sizes look in a minute, it stopped working. And so what was going on? Were we wrong? Were these all file drawer effects? Were they false positives? Well, there was 2 things that came out of this. One, in Adam Guastella's study, what they found is people who got the drug but did not get better within session. Well, so other way around. If they had good within session extinction and they got the drug, they got much better. If they did not have within session extinction and they had the drug, they either got worse or got no better. And then Barry Everett, one of the leaders in preclinical models of learning memory, explicitly tested this. They said, well, perhaps reconsolidation is also NMDA dependent. And they explicitly tested, and they found that D-cycloserine potentiated both the extinction and the reconsolidation of fear, depending on the length of the extinction memory reactivation session. So the take-home message is now we think that every time a new memory comes up, different memory traces get activated. If it comes up briefly and you leave it, it activates that memory trace, NMDA activation, and that is strengthened. If you come up and you stay there for a long time, that's enough time for essentially the error signal will come back through, work back through the system and start to retrain the brain that extinction will occur. And so as that has been taken into account, there's been a number of positive studies depending on how you dose it, when you dose it, and how you're sure that there's exposure therapy. The important point for me is we're all excited about ketamine and psilocybin and plasticity-mediated modulators, which again have these amazing effect sizes that look a little too good to be true. And my worry is, and this is just with Adriana Feder, who's doing great work showing exposure therapy plus ketamine, but that if we're not careful, that all of these plasticity mediating agents can likely both increase reconsolidation or increase extinction if we're not controlling for the exposure. So I want to end then on clinical ways that maybe we could even do better in the future. And the question is, instead of enhancing plasticity, but having no control really over whether we're enhancing reconsolidation versus enhancing extinction, if these are truly different circuits that mediate extinction, could we only enhance plasticity on the extinction circuit and not the reconsolidation circuit? And it might be possible. So Andreas Luthi showed us about 15 years ago, that there are different cell microcircuits in the amygdala that mediate fear on or reconsolidation versus extinction. So they recorded with single units in mouse amygdala during tone shock, pairing, and extinction. And they found that about 30% of the cells didn't respond to the tone at baseline, but after tone shock fear conditioning did respond to the tone, but then after extinction, they didn't care about the tone anymore. So those mimicked the behavior. Then they found these that they called extinction or fear off neurons. They didn't care about the tone at baseline. They didn't care about the tone with fear conditioning, but after fear extinction, now they cared about the tone, as if they were holding that tone fear memory inhibition. I've talked about the amygdala. We really know there's about 15 subnuclei, and now we know there's hundreds of different cells and microcircuits, but we're now starting to have molecular control over this. And I just want to end on one story of a set of neurons in the amygdala marked by the Thi1 marker. And it turns out the CAM kinase positive basolateral neurons that project out of the BLA are made up of different subtypes. And about 30% of those subtypes are marked by the Thi1, which come from a cortical development population. Interestingly, the canonical theory is that the basolateral amygdala projects to the central, and that leads to the hardwired fight or flight response. But none of these green cells are projecting to the central amygdala. In fact, these Thi1 cells project preferentially to the nucleus accumbens and to the infralimbic more than the prelimbic. And if you now do in vivo physiology, and this was with Tig Rainey at Emory, if you activate the lateral amygdala circuit coming from sensory systems and measure central amygdala outputs, and you optogenetically activate these basolateral amygdalins, you actually block the flow through of the canonical fear circuit. Behaviorally, if you activate these with genetics, activate these putative fear off cells, you acquire fear just fine and you completely block what we think is the consolidation of fear, or if you activate them during extinction, you get a robust enhancement of extinction. If you use chemogenetics, you can do the exact same thing. In this case, we're activating the fear off cells, and in red, we block fear. If we do a GI, we get more fear. So Ken McCullough in our group wanted to say, well, could we have other markers of these specific Thy1 fear off cells? So we did FACS sorting of the Thy1 mark cells and found a bunch of genes, some of which, like RSPO2, Suma Tonagawa's lab had also shown might be involved differentially in the amygdala to accumbens projection. And the important point is we found a number of new targets. In this case, neurotensin 2 receptor was preferentially expressed in these fear off cells compared to the neighboring cells. Neurotensin 2 receptor is a GS-coupled stimulatory receptor. We knew nothing about it, but we got an agonist for it. And sure enough, when we use an agonist that activates the fear off cells, we got this amazing blockade of fear expression and enhancement of extinction. So it's proof of concept that we're now, and then this work was Kay Tai's group, and we collaborated with a recent Nature paper showing that neurotensin modulates positive and negative valence in the amygdala. So the take-home message is, I think the field is far enough along in understanding the cell type molecular mechanisms of some of these fear on versus fear off pathways. We can start to have a targeted pharmacology of fear. So in summary, I think we're to the point that we're starting to have a number of new pathways based on the neurobiology that we might be able to give at the time of extinction or even at the time of trauma exposure to block fear consolidation, leading to a novel neuroscience-driven intervention. The second summary is I reminded you of we can break down the development of PTSD to multiple different compartments, and to remember that it's got genetic and environmental components of early risk, and consolidation is something we can manipulate. But by understanding processes of extinction and discrimination instead of generalization and sensitization, it can lead to novel targets. The most simple summary is PTSD is essentially the amygdala has gone off the rails. It's responding in a fearful way all the time. And the brakes of the amygdala and prefrontal cortex have fallen off. Our trauma-informed CBTs increase top-down regulation through essentially training the brain. Our current treatments essentially just sort of dampen the symptoms of anxiety and depression, but they're not getting at the underlying course. We hope that future medications based on neurobiology will allow us to target these circuits. And I'm not going to go through all of this, but we believe that based on a whole bunch of approaches, that PTSD and fear and anxiety are a tractable set of disorders, and that they may lead to a precision medicine approach to trauma and psychiatric disorders based on an understanding of neurogenetics. So I tried to thank everybody as we went. It's been a huge team that I've worked with over the decades, and thank my funding sources, primarily NIH. And thank you for your attention.
We have some time for questions, and I'm hoping that people will write them in. And as you enter your questions online, I'm wondering if anybody in the room has any specific questions. I know I have one I'll ask right now. I was planning to ask you about synergy between medications or biological interventions and therapy, and I didn't have to ask because you addressed it so well. But let me ask you, do you think there's any role for transcranial magnetic stimulation, which can have the temporal precision, perhaps, that we might be lacking with medications? And is anybody studying that?
Yeah, so there's interesting studies saying with TMS, where you essentially summary. Thank you, Lydia. That's slide 2. That with TMS, you're enhancing plasticity, but perhaps you're enhancing plasticity in relatively specific circuits. And the most common is DLPFC. And we think that some of the Medectin's work suggests that DLPFC activation actually inhibits amygdala activation. So there are studies looking at using written exposure therapy combined with TMS. We actually had a study that we were very excited about that was a very robust negative in which we used the BrainsWay H coil to target what we thought was the infralimbic by or the subgenual cortex by having a medial target. And the thought was that would enhance the extinction circuit while giving written exposure therapy. In fact, people who got written exposure therapy alone did better. Those who got, and with sham, those who got the TMS actually got not markedly worse, but not nearly as well as those who got the sham. So it was a true negative result. And what we think happened is either A, that the coil activation didn't go deep enough when we were actually activating the dorsal anterior cingulate, which generally goes with the amygdala as opposed to the subgenual, or that we were getting more of an inhibitory TMS burst than an excitatory TMS burst. So it remains to be seen, but it suggests that all TMS is not equal. We kind of know that. Where you hit the circuit is going to matter, but as we have a better understanding of these circuits and combine them with learning processes, we might be able to really do better.
Well, not only is rapid and slow TMS potentially going to discriminate some of those, but what about theta burst? I wonder.
Yeah, so I think that is the question, and I think it's partly of, do we understand enough about theta burst and accelerated TMS and plasticity? If it's doing what we're thinking it's doing, we should be able to have the temporal component of enhanced plasticity with theta burst with exposure therapy and extinction, but we probably still have this caveat of we need to make sure when you're doing the exposure that it's a full exposure and that we're not potentially getting a reconsolidation event.
Right. Thanks.
It looks like we do have a couple of questions. So Kat, hey, Kat Liang out there in the web world. Kat says, folks have been studying cortisol and mental health for a long time, but it hasn't done much with our current treatments. The work with FKBP5 may lead to a renewed enthusiasm with glucocorticoids. Wondering if we can comment on where we envision the future of this area going and strategies to overcome it. So John and I had this exact conversation, I guess, an hour and a half ago. And one of the things I wonder about, and I think we need to be thoughtful about, is I think we all know who've been around for a while that typically dirty drugs work better than clean drugs, right? Our tricyclics and our MOA seem more powerful, as well as our clozarils than our very specific antipsychotics, right? We know in other areas of medicine, be it oncology or infectious disease, rarely does monotherapy work. The systems are generally smart and they come up with compensatory and they overcome the drugs, whether it's resistance or whatever. Well, the brain is the most plastic thing in the universe, as far as we understand. Is it a wonder that as we better understand specific neurobiology circuits and we're targeting them very specifically, that the brain doesn't have a way around this? So I do wonder if we need to be, and then I think you could take the same question about cortisol modulation with the CRH antagonists, which were one of the first drugs that were safe and you could be targeted these peptides, and yet it was pretty negative. But maybe we need to take the CRH antagonist plus the FKBP5 antagonist, et cetera. Maybe we need to be really using our understanding of neurobiology to hit these circuits at more than one place to maximize their effect. So that's where I hope the future is going, Kat, but I think we have to do more research to figure that out. But I think it's a combination of precision medicine. We're treating everybody with depression or PTSD the same when we know there's biological differences. So we have to get those biological subtypes, but we probably also have to hit these circuits more than one way. Another question, can you please share the study or reference of the NMDA study in which NMDA activated extinction and reconsolidation? I will just back up and leave it on the screen for a minute. It's Barry Everett. It's right here. It was J Neuroscience. If you search this, if you want to send me an email, I'll send you the specific reference. I don't remember the exact year. Any thoughts on why chronic PTSD would make memory poorer in aging people? I think, hey, Eric, good to see your name up there. The simplest answer is that PTSD makes cognition worse for everybody, because essentially you're driving your limbic system survival reflex over your higher cortical and hippocampal declarative memory systems. And we have to remember we're all the time have multiple memory systems in parallel, and we generally think of memory, when we say memory, we think of hippocampal declarative memory in facts and places. But emotional memory systems are probably more like motor memory systems. We don't have as much conscious awareness of them, but they're happening in parallel and they're stronger. And when they take over, the rest of the system doesn't work as well. So that's one example. In aging, we know that chronic PTSD increases risk for dementia. It increases inflammation. It increases oxidative stress. So for all the reasons that we see probably also decreased memory in any of our chronic psychiatric disorders, I think we're also going to see it with chronic PTSD.
I have one more
Yes,
question.
please, Matt.
So it seems like the symptoms of PTSD have kind of this intermittent component with like the re-experiencing, and that's what you're kind of targeting with the memories, right? It's like having the memory of the bad event. We want to kind of extinct that sort of thing that happens intermittently. But then there's also kind of these symptoms that occur more kind of continuously, where like someone's just on edge or anxious. And so I'm wondering about like, have you thought about the different sort of are those the same neural circuitry being
Yeah.
targeted in both cases, or do you feel like there's because I know a lot of your work is on like general
Yeah.
fear and anxiety
Yeah.
in general as well.
So the simplest answer, which may or may not be right, but at least there's some data, is that, and what Mike Davis would say probably is that the BNST, Beta-nucleus of the striatum terminalis, is, so when I say the amygdala, I've simplified it as the basolateral. Of course, there's lots of reasons, and now we talk about the extended amygdala. And the BNST is a region slightly anterior and central to the amygdala, which has very many of the same inputs and very many of the same outputs. And so David Walker and Mike Davis's lab showed in the early 90s or early 2000s that BNST seemed to moderate more distal fears and distal threats and unpredictable threats, and the basolateral more direct, immediate cues. And so the thought's really been that these two are kind of working in parallel, but the more generalized anxiety or unpredictable stuff may be more BNST driven, and the more direct threat and triggers is more amygdala driven. There's some of the human BNST imaging is consistent with that as well.
I think I'm going to take this pause to thank Kerry for his presentation today, and to emphasize for our trainees how inspiring it is to see you learning from the patients that you were taking care of, and then turning that into research that hopefully will drive forward new treatments. And that's something we should all keep in mind as a paradigm that we should pursue.
Yeah.
So thank you very much, Kerry.
Wonderful career. Thank you so much. Thanks for your attention. Bye, everyone.